Hi, welcome everyone. Today we're going to be talking about the physics, biology, and biochemistry of photosynthesis. Let me just give you a big thank you from the bottom of my heart. I'm glad that you guys want to stay and take a deeper dive into photosynthesis. I promise I'm not going to get very deep into all of these concepts, but I want to provide you with enough information to understand what is going on and maybe help you if you have any exams or you just want to win a fun trivia battle because we know sometimes we like to be smart. So where exactly does photosynthesis happen? If you guess the leaves of plants, you are correct. So if we zoom in, they seem like paper thin, almost like nothing's going on, but on the microscopic level, there's a lot of order to leaves of plants, and it's actually pretty thick. And so if we zoom in, we have like these middle cells here that are in between the top and bottom layer. We have a plant cell. We can see what these little dot structures are within the leaf. And if we zoom in on one of those, these are called chloroplasts, and they're a double layered organelle. And this is showing what it looks like in real life. Now, if we look inside of the bean, we can see a bunch of like structures inside of it. And each one of these tiny little discs here are called thylakoids. This is important because this is where the light-dependent reactions occur. Outside of these tiny discs, in this space in between, it's like a fluid-like substance. It's called the stroma, and this is where the light-independent reactions occur. Now you may see there's like stacks of discs here. Each one of those are called granum. Now don't get that confused with your grandma. She's not a plant or part of a plant. They're called granum. Now that you know all the parts of photosynthesis, let's talk about light. What is it? How do plants harness it? And why do they spend so much time and energy doing so? Before we can start answering some of those questions, let's understand what light is on a fundamental level. The same light that you use to make sure there aren't any monsters in your room, or the light that is from your phone screen, is also the same light being emanated from the sun. But what is your phone screen, light bulbs, and sun emanating? Well, over on the left here, this is our best representation of what a single light ray or photon looks like. And I know what you're thinking. What am I looking at? And that is such a great question. See, light doesn't have a mass. Therefore, it is a form of energy. Specifically, it's the movement of energy in both the electric and magnetic fields. Now, this energy has specific parameters that qualify as light and not any form of energy, but for simplicity's sake, we're not going to get into that in this video. This light energy, however, is known as electromagnetic radiation. And before you ask, no, the light is not radioactive. Radiation just refers to something being emitted or moving away from a source. It's emitting or it's radiating at the speed of light. And each light ray creates a specific pattern within the electromagnetic field, creating a wave pattern. Now, the distance between two crests or two peaks of this wave pattern is known as its wavelength, and it's equal in both the electric and the magnetic fields. And the number of times each peak occurs per unit time is known as its frequency. And now you can imagine they're going to be very large and very small wavelengths, and that is true. And that is what the electromagnetic spectrum is. It's the total possible range for electromagnetic radiation, and it's divided into a couple different bands. So if we look down at these two figures here, if we look at this figure first, we can see the bands are grouped based on its wavelength or its frequency. The higher the wavelength, the lower the frequency. The lower the wavelength, the higher the frequency. They're inversely related. Also, the lower the wavelength, the higher the energy. The larger the wavelength, the smaller the energy. That is also inversely related. But these bands, starting here, high energy, gamma rays, then they move on to X-rays. These are the same X-rays you use in a hospital. Then there's UV light. This is the same UV light as in black light or that burns your skin. Then you have this little band here called the visual spectrum. This is all the colors humans can see. Then we move down and we have infrared light. Then we have microwaves, the same microwaves that your microwave uses. Then we have two classifications of radio waves. We have short radio waves, and then we have long radio waves. At these distances, the wavelength of these radio waves are the size of cities. I know that was a lot, and I promise this will all come back together in upcoming slides, but I want to zoom in on visible light. So if we're going to look at this figure here, it better explains what I've just said. It's also sorry that they're inverted. You have violet on the right here and then violet on the left. Typically it looks this way, but sometimes it's inverted, but they're saying the same thing. So the larger the wavelength, the less energy, and it's going to look like this. Each color has a specific pattern and energy. As you move up, the wavelength is going to get shorter and is going to increase energy. And typically, visible light ranges from 380 to 750 nanometers. Lastly, true white light is the combination of all visible light together. Now that we know what light is, let's learn how plants use it in order to perform photosynthesis. As you guys may know, atoms are the fundamental building block of all matter, and they are comprised of a nucleus, which has protons and neutrons, and they're surrounded by tiny negatively charged molecules called electrons. And just like how a ball, or let's say a watermelon, prefers to be in the ground, where it has zero chance of falling, whether that be in the air or let's say off a table, electrons also prefer to be in their lowest state, also known as the ground state. Because if they were to fall, the watermelon would have to break, and your whole family would be angry because now you have to get a new one, and that definitely was a made-up hypothetical and something that did not happen in my childhood. Yeah. However, on a real note, remember that light is electromagnetic radiation. Well, if the photon were to hit an electron, the energy from the photon is going to transfer from there and essentially energize the electron. When this happens, it creates a high energy electron. And this space here, from light hitting the electron and making it excited, making it a high energy electron, popping it into a new orbital, this process is the key to understanding photosynthesis. But we will get into this in the further slides. For now, let's look at the specific wavelengths plants use for photosynthesis and why they do. To understand this, we have to look at a special molecule called chlorophyll. In the plant world, chlorophyll is the main pigment molecule used to capture solar energy. There are two main types of chlorophyll. There's chlorophyll A and there's chlorophyll B. But what's the difference? Well, if we look at this absorption spectrum, what we're looking at is the different wavelengths of light or color of light versus the amount of light is absorbed, or the amount of light energy that is converted into high energy electrons. We can see in the light green is the chlorophyll A absorption spectrum, and the dark green is the chlorophyll B absorption spectrum. Chlorophyll A likes to absorb violet light and red light. Chlorophyll B likes to absorb blue light and orange light. It's in the plant's best interest to absorb all the different types of light that are safely possible. This way, the plant is maximizing photosynthesis. It's photosynthesis maxing. And this is done by other pigments within the plant, such as carotenoids. These pigments absorb light that chlorophyll does not. For the example of the carotenoids, they absorb mainly light blue and green wavelengths. But we have other ones that absorb yellow ones and orange. Also, there's these other molecules that make the leaves appear different in pretty colors in autumn. They need high concentrations of sugar for that to occur. But to understand why leaves are green and not any of these other pretty colors that we see and love during autumn, we have to look at the right side of this figure. This is showing the action spectrum of photosynthesis. Again, based on the wavelength or color of light, we can see what the rate of photosynthesis, the highest peaks of photosynthesis occur around the violet and blues and the orange and reds. And this correlates very heavily with the chlorophyll molecules. And because of this, plants heavily invest in these molecules. But the final piece into answering why leaves are green and not any of the other colors lies in the gap between these two peaks and a little bit of physics. You see, the vast majority of matter in the universe, if light is not absorbed, then it's typically reflected in some kind of way. In this case, green light is the main light not absorbed, okay? plants are not absorbing this green light. Therefore, the light is reflected back to us. And that's why leaves appear green. And you might ask, why don't plants absorb this? I get why chlorophyll absorbs this and why you have 2 peaks, but why isn't there more molecules to absorb green light? And to answer that, to make that very complex question and simplify it in a great answer for you, is because plants want to balance low energy and high energy. If you get a balanced state of low and high energy, there's no need to absorb medium energy, and it's safer for the plant to do this as well. If you want to learn more, please leave some comments and maybe I can answer you more specifically why they do this. Fairly complex, but it's kind of cool. We're not getting into that though. However, the concept of reflection works for every other matter in the universe. For example, snow appears white because it reflects all white light. The orange iPhone 17, orange leaves, or oranges appear orange because they absorb all of the colors besides orange. They're reflecting orange light. In the case of black objects, they're absorbing all visible light and they're reflecting light that we cannot see, but our eyes interpret that as black. Now that we know the basic physics of light and how it is captured, let's see how plants harness it and use it in the first half of photosynthesis. So, the first half of photosynthesis is known as the light-independent reactions, and this is where light is harnessed and used within the entire process of photosynthesis. In short, this figure here illustrates the entirety of the light-independent reaction. More specifically, it's showing a series of proteins that captures and use the light energy to perform work, also known as the electron transport chain. Some key protein complexes are Photosystem 2, the cryochrome complex, Photosystem 1, NADP plus reductase, and of course, ATP synthase. This protein is arguably one of the most important proteins in the entire cell. More on that later. But let's zoom in on one of these photosystems. Over in this figure, it is showing a great illustration of the basic components of both photosystems. They are comprised of light harvesting complexes and a reaction center. As the name suggests, the light harvesting complex is where light is captured. This is also where all the pigment molecules are stored within the cells. So, photons, light, is going to hit these pigment molecules, and the energy from here is going to be absorbed. The absorbed energy is going to transfer from pigment molecule to molecule and make its way down to the reaction center. There, the energy is then going to be transferred to a specific pair of chlorophyll A molecule. This specific pair of electrons, once both are energized, are going to move to the acceptor slot and then be transported along the electron transport chain. And that is in essence how photosystems work. But each photosystem is a little bit different. This system starts with water molecules and photosystem 2. Now, before we get any further, photosystem 2 is called photosystem 2 because it was discovered second. Photosystem 1 was discovered first, but the second one is the first in our chain. Got it? Get it? Good. At the reaction center, it splits one water molecule, creating 1/2 of an oxygen molecule and 2 hydrogen ions. Then it's repeated again to eventually generate the 2 high energy electrons, meaning it takes 2 water molecules to create 1 oxygen molecule and 4 hydrogen ions that then accumulate and stay within the thylakoid space. The oxygen molecule is then released as a byproduct through the cell and through the leaf because the leaf doesn't need it at all. Okay, before we get any further, let me take a step back. Because of photosystem 2 and this entire process of photosynthesis, we have enough oxygen for all living creatures on Earth to breathe. So photosynthesis and having enough plants alive and planted is critically vital for billions of species survival, okay? And exponentially more important to have environmental regulations in place that, one, conserve and protect the plants that we already have, and 2, put in new laws so that for all future construction projects, they implement more plants in and within the projects, including on buildings, essentially creating the same environmental laws that the country Singapore has, where they truly live with the land and not against her. Okay, electrons that are then energized are going to move along this complex. They're going to be transferred to the cryochrome complex, where the electron's energy is spent to bring in more hydrogen into the thylakoid space. The spent electrons are then going to be moved to photosystem 2, where they're re-energized by light. They're going to be transferred to NADP plus reductase. There, the energized electrons are going to be combined with NADP plus and a hydrogen ion to create a single NADPH molecule that then move on into the light-independent reactions. Okay, we're almost done, I promise. Okay, you might be asking, why do plants heavily invest in creating a very acidic or high hydrogen ion concentration within the thylakoid space? And that is because it is needed to run arguably one of the most important protein complexes within the cell, and that is the ATP Synthase complex. It does this in a similar way that a dam works. So a dam needs a large reservoir of water behind the generator in order for it to flow down and through the generator, generating energy before it gets to a lower concentration. This is essentially how ATP synthase works. The high concentration of hydrogen ions naturally want to flow to the lower state. ATP synthase takes advantage of this natural concentration gradient to combine ADP molecules and a phosphate group to create ATP. And this is what the ATP synthase complex looks like. Sorry, it's upside down from this figure up here, but this bulb area is this, and this area over here that's inside the thylakoid is this area down here. So hydrogens are going to flow in through it. It's like a turbine. It turns it, and then ADP plus a phosphate group is going to be combined, and then ATP is going to be put out. I don't have the time to get into more of this fascinating protein, but if you do, and this I highly suggest watching the Harvard University's video on this protein. The link for it will be in the description. And if you are curious, this entire reaction equation for the light-independent reactions, this is what it takes to generate a single glucose molecule. And with that, let's move on to the second half of photosynthesis. And that is known as the light-independent reactions. It gets its name because the reactions occur in the presence or absence of light hitting the leaf, meaning it occurs during the nighttime or the daytime. It doesn't matter when. And before you get scared looking at this next figure, I promise, I'm going to spare you the biochemistry, okay? But what you're looking at is the light-independent reaction, also known as the Calvin cycle. It occurs in 3 main steps: carbon fixation, reduction, and regeneration. And before I get any further, the numbers associated here, you have 3 CO2, 3 RuBP, 1 G3P. These numbers here are going to be associated with the cycle occurring 3 times. Okay? But first, let's talk about step 1, carbon fixation. Here, carbon dioxide molecules from the atmosphere are going to enter the cell and make their way to the stroma of the chloroplasts. There, CO2 is going to be combined with a molecule of RuBP from the help of this enzyme called RuBisCo. From there, 2 3-PGA molecules are going to be created. These 3-PGA molecules then enter the reduction stage. There, these molecules are transformed by the help of ATP and NADPH that was already generated to create G3P molecules, where most of these molecules are going to move on to the regeneration stage. This is where G3P molecules are converted back into RuBP. Now, like I said, the numbers here occur for 3 cycles. So, after the 3rd cycle, 5 G3P molecules are going to be regenerated, which leaves only 1 G3P molecule to move on to make glucose. So, after 6 cycles, 2 G3P molecules are going to be moved out of the chloroplast to make glucose, and 10 G3P molecules are going to be used for regeneration. In an idealistic world, this is what would happen every single time. but unfortunately that is not the case. That is because of the very important enzyme that captures CO2 from the atmosphere, called RuBisCo. An enzyme is supposed to speed up the natural process of reactions, where many metabolic enzymes have turnover rates of over 1,000 reactions per second, whereas RuBisCo only averages 3 to 10 reactions per second, making it significantly slower than many other enzymes. But wait, there's more. On top of this, RuBisCo likes to, how shall we say, make mistakes. And instead of grabbing CO2, it'll grab an oxygen molecule instead. This is going to shut down the entire process and cost the cell 1/3 of its energy generated just to fix. Evolution decided that instead of creating a more efficient RuBisCo, they would just invest in creating tons of RuBisCo instead, thus allowing the benefits to outweigh the negatives, and making it single-handedly the most abundant protein on Earth, and is also presumed to be why most G3Ps are regenerated instead of moving on to form glucose. Here's a kicker to all of this. You see, at a certain point, RuBisCo's problems increase as the temperature increases outside, meaning on top of the numerous problems that global warming is going to cause, it is also going to cause photosynthetic rates to go down. So as we see in temperature here, technically as temperature goes up for the beginning part, photosynthesis is actually going to increase. But once you get past your 35 to 40, it's the most idealistic range to be to have photosynthesis occur. However, after 40, the threshold for photosynthesis to stop functioning properly dramatically decreases and occurs rapidly. So we want to avoid this idealistic range because proteins are actually going to denature and not actually going to work properly. So we don't want temperature to increase outside. This is going to create a negative feedback loop where the increase in CO2 concentrations are going to increase temperature, which then increase the problems with RuBisCo, which means less CO2 is going to be captured, which then increases CO2 outside, increasing the temperature again. So it is critical to our species survival that we lower and reduce the amount of human-derived greenhouse gases, because there will become a point where plants will no longer be able to photosynthesize, which will end most life on Earth, period. Some researchers are aware of this and are trying to genetically increase the efficiency of RuBisCo in hopes of slowing down the effects of global warming on photosynthetic rates in all plants. If you want to learn more, go check out the other video in my description. With all of that being said though, why are plants heavily investing in glucose? After all, it is a very inexpensive molecule to make, requiring 18 molecules of ATP and 12 molecules of NADPH to create a single glucose molecule. A better question is, what isn't glucose used for? You see, glucose is involved in a large variety of cellular processes. Some notable ones is in the creation of energy, where it averages 32 ATP molecules per glucose, though the theoretical yield is slightly higher. It is also involved in cellular growth, where it is converted into cellulose, which helps the plant hold their shape. It's also involved in the creation of nucleic acids like DNA and RNA. It is also involved in the creation of proteins when combined with from nitrates from the soil. So you see, glucose is very, very important to the cell. Although I do have to say, photosynthesis ends technically at the creation of G3P molecules leaving the cell, 2 of them specifically. These G3P molecules are going to leave the chloroplasts and enter gluconeogenesis pathway. There, G3P molecules are going to be combined with abundant enzymes and water, and then glucose is going to be created. If you're curious, this is what light-independent reactions look like to create a single glucose molecule. But again, photosynthesis ends at the creation of 2 G3Ps because it does not create glucose. That's where gluconeogenesis comes in. Taking everything that we have learned, I thought it'd be nice to have a full overview of what photosynthesis actually is. As with most things in science, we have to make complex things simple, and photosynthesis is no exception. It is true that photosynthesis is the overall process that converts sunlight into sugar. But from what we have learned today, it is much more complex than that, or rather, in order to perform that simple yet vitally important process, it requires feats of molecular evolution in the form of organelles, proteins, and many production problem solving. A more accurate way to describe photosynthesis is this. Photosynthesis is the overall process that occurs within the chloroplasts converting sunlight into glucose. It encompasses how blue and red electromagnetic radiation and water are absorbed within the thylakoid membrane, generating high energy electrons that perform work and also generate ATP and NADPH, where oxygen is generated and released as a byproduct. Photosynthesis also encompasses the fixation of atmospheric CO2 within the stroma by the enzyme called RuBisCo. The resulting byproduct, 3-PGA, is combined with the previously generated ATP and NADPH to generate two molecules of G3P that move out of the chloroplast in order for gluconeogenesis to occur, and 10 molecules that are recycled for future light-independent reactions. If you're curious, this is the full reaction equation for photosynthesis. This is for the light-dependent reactions, and this is for the light-independent reactions. Again, this whole process is leading to the creation of two specific molecules, the first of which, the golden egg, is a single glucose molecule. And if you weren't aware, this is what it actually looks like within the cell. This is known as a chair conformation, and it shows the overall 3D structure of the glucose. You might see some other projections, and they're all correct. However, they take away from what it actually looks like in order to help other things instead. So if we set this down horizontally and we look at it from the sky vertically, you're actually going to see a ring structure, a hexagon, and you might be familiar with glucose looks like that way. Although glucose is used in a large variety of processes, one key creation is in this molecule, which is an ATP molecule. And specifically, it's the breaking of these bonds here from the O to the P that generate energy for countless operations. Okay, before I let you go, remember, without this process, photosynthesis, oxygen would not be released in significant quantities for all life to use. In addition, photosynthesis acts as a natural carbon sink, storing atmospheric carbon dioxide. So it is vital for our species' survival to protect the vegetation on Earth that we have and introduce more vegetation and regulation on future projects so that we can start working and living with the land and not against her. Alrighty, that's all I have for you today. Thank you for watching and wanting to learn more about photosynthesis. If you have any questions, comments, concerns, please leave them down below. And if you like the video and want to support the channel, consider liking and maybe even subscribing. That way, the information can reach the people who actually need it. Please go on and check those videos down below and know that I love you all. Here are all my sources in case you wanted to learn more about any specific topic.